Hello, good day everyone. Today we're going to discuss about cell cycle and cell division. So you already know that your body is made up of cells. As a multicellular organism, these cells make up tissues, which will consequently make up organs and organ systems. So organisms, like plants and animals, rely on this complex organization for the proper functioning of their bodies. However, any sexually reproducing multicellular organisms begins at a single cell as a single-celled entity. This cell then relies on cellular events to produce more cells, the early stage of which is known as the blastula. As you can see in the picture, that's what you call the blastula. Further growth and development proceed through the continuous reproduction and specialization of cells. So the process repeats for as long as needed to ensure the maintenance and survival of an individual. Although not a perpetual process, organisms may still rely on the production of new cells on some occasions. So we have discussed on our previous lesson that the cell undergoes cell division, which is your mitosis and your meiosis. So how do our bodies replenish a cell? So have you ever wondered how our bodies replenish cells? With very few exceptions, all of the cells in our body contain genetic material in the form of DNA or your deoxyribonucleic acid, which is found in the nucleus of your cells. So DNA is an extremely important molecule because it contains the information that codes for your traits and the processes that ensure survival. It does so because of its notable structure, which is made up of two helical strands of repeating units called your nucleotides. These nucleotides form base pairs, the varying sequences of which code for different traits. Most of your biological traits are actually coded for by DNA. Your eye, or the color of your eye shape, your hair color, height, and many other heritable traits that are coded for by the DNA. DNA has many forms by which it appears in the cell. The phase of the cell cycle usually dictates what form DNA will appear as. DNA is normally found in the form of chromatin while it is inside the intact nucleus of the cell. So DNA as a molecule is very long. So what we have, as what I have mentioned in our previous lessons, it measured around 6 feet. So this is why it needs to be packaged into a form that can fit inside the cell. So this is done through the basic structure known as the nucleosome. So the nucleosome refers to the level of DNA packaging where a segment of DNA is wrapped around a positively charged proteins known as histones. Collectively, all the DNA molecules of a cell constitute its genome. When the cell is in the phase of the cell cycle known as the interphase, which will be discussed later on, then the DNA is usually found in the form of chromatin. So the chromatin, or the conformation of the DNA that is observable during interphase, is a complex of DNA and histone proteins. Histone proteins, where the DNA molecule is wrapped around, helps organize the genetic material inside the nucleus. Chromatin will sometimes need to further condense into a highly coiled and compact structure. This highly condensed form of DNA is known as a chromosome, as shown later on. The condensation of the chromatin into the chromosome usually occurs during the cell cycle, when the cell is about to divide, which will also be discussed later on. Diploid organisms have chromosomes that occur in pairs in each of their cells. So each pair of chromosomes are highly similar to each other, but still bear a few differences. These pair of chromosomes are known as homologous chromosomes. For example, Humans have 46 chromosomes in their diploid number. So these chromosomes can be arranged into homologous pairs for a total of 23 pairs. So each duplicated chromosome contains two identical members, each member of the duplicated chromosome known as a chromatid. It contains the exact same genetic material as the other member. So the two chromatids having the exact same genetic material due to DNA replication are then known as the sister chromatids. So each sister chromatid in a chromosome is a link to the other chromatid via the centromere. Now, to summarize it, cells contain genetic information in the form of a double helical molecule known as your DNA or your deoxyribonucleic acid. Segments of your DNA are wrapped around proteins called your histones, and the resulting complex is known as nucleosome. So further coiling and condensation of the structure result in the formation of chromosomes, which are essential during your cell division. Now, let's try to answer this one. So you are looking at a diploid cell which consists of 4 pairs of chromosomes. After DNA replication, how many chromatids are there? How many of these do you think are homologous? Now, you try to answer on your own this question. The chromatin condenses into chromosomes. Each half of the chromosome is a chromatid. Now, to help you remember this, imagine the chromatin to be a very thin string of DNA. So imagine the string being wound up into a thick rope. So this thick rope is the chromosome. Now, imagine each half of the rope to be similar to the other half. These halves are what we call chromatids. Now let us proceed to the introduction of your cell cycle. Let us recall how different organisms that you know grow and develop. So plants produce seeds that sprout into seedlings. So these seedlings grow and mature and produce more seeds in order to produce more plants. So frogs, let's say the frogs, on the other hand, they lay eggs. So these eggs hatch into tadpoles, which metamorphosed or change into froglets that eventually grow into adult frogs. These adult frogs will then lay eggs that will hatch into new tadpoles. Us, or you as a human, were born as an infant, grew into a child, are now a teenager, and will eventually become an adult. Some species of organisms have their own life cycle that shows how they are born and how they are developed and how they are matured. Much like entire organisms, individual cells also have their own life cycle. That very single cell has its own life cycle. So the life cycle of a cell is known as the cell cycle, and this describes how cells grow, develop. So what are the different phases of the cell cycle, and how does the cell make sure that these phases proceed smoothly? The cell cycle is a process that is essential to the growth and development of cells. So in addition, it ultimately leads to the reproduction of cells, similar to the life cycle of different organisms that occur in a stepwise fashion. That only means to say it follows a process. Different phases also make up the cell cycle. So each of these phases will have specific events that contribute to the overall maintenance and survival of the cell. So what are these phases of the cell cycle, or the stages of the cell cycle? So we mentioned earlier that the cycle is made up of multiple stages. So each of these stages will have specific events and occurrences that are crucial to help the cell function properly and be prepared for the division. So there are three main stages of the cell cycle, namely its interphase, the M phase, and your cytokinesis. So let's proceed to your phases of your cell cycle. So most cells spend the majority of their time in the stage known as your interphase. So this stage is when the cell prepares itself for eventual cell division, which is the process of how cells produce more cells. So during your interphase, the major events that occur include the growth of the cell and the duplication of its genetic material. So your interphase can be further subdivided into G1, your S, and your G2 phases. Now, for gap1 or your G1 phase, the first stage of your interphase is the G1 phase or your gap1 phase. So G phase are considered gap phase because they intervene between DNA replication and cell division. G1 phase usually begins as soon as the previous cell cycle ends with the newly produced daughter cells. So this phase involves the growth of the cell's cytoplasms alongside the doubling of cellular organelles. Proteins and other molecules are produced for normal cell functioning. So it is also during this stage that the rate of the synthesis of proteins is the highest. So other products that are produced include your enzyme, your nutrients, and energy molecules. A structure known as the centrosome will eventually be important during cell division. So these centrosomes are composed of centrioles in animals. So these are cytoskeletal elements that will be crucial in distributing the chromosome into their respective daughter cells. So we have mentioned in our discussion of the cell parts and function that the microtubule or the centrioles, the microtubular structure, which are your centrioles, are responsible for the assembling or for the production of your spindle fiber that will be used by the cell during the cell division. So during the G1 phase, the centrioles of the centrosomes move away to await the assist the events of mitosis. If all of the conditions of G1 checkpoint are satisfied, the S phase or the synthesis phase takes place. So this stage is named so because it is when the cell synthesizes a copy of its DNA in a very notable process called DNA replication. So this refers to the event when an existing DNA produces another copy of itself. Through this process, the cell can effectively and almost accurately duplicate the genetic material that it has. So the duplication of the genetic material is very important in fulfilling the objectives of the cell cycle. So let us recall that mitosis aims to produce daughter cells that will contain the exact same copy of genetic material as the parent cells. So this cannot occur if the parent cell only has one copy of the genetic material. For the daughter cells to have identical copies, the parent cell will need to duplicate their own genetic material. So this is made possible by DNA replication with a very minimal errors during the process. The next event in your interphase is your gap2 phase. So after DNA is replicated in the S phase, the cell will then enter the gap 2 phase or your G2 phase. So this is the second gap period between the replication and the cell division. So important processes that happen during this phase are continued growth and the production of materials that are necessary for the cell division to occur. So proteins are also synthesized during this period, but not at the rate similar to that of the G1 phase. So after the cell exits interphase, it should now have all the factors necessary for mitosis or meiosis to occur. So the phases involved in mitosis and meiosis will be discussed in details later on. Okay. So once the cell is sufficiently prepared to divide, the division of the nucleus, which includes the duplicated genetic material, will now occur. Now, this event in eukaryotic organisms involves either one or two processes, namely your mitosis and your meiosis. So these forms of cell division differ with the type of cycle or with the type of the cell. Example of which is your sex cells or gametes that undergo meiosis, whereas the somatic cells or your body cells or non-sex cells undergo mitosis. So the goal of mitosis and meiosis is to divide the nucleus of the parent cell and distribute the genetic material to the resulting new cells. So mitosis and meiosis have several phases, and each of these phases has specific events that contribute to the proper partitioning of the genetic material. The phases of mitosis and meiosis will later be discussed. Now, once the cell division concludes, then cytokinesis will occur, as you can see in this image. So cytokinesis refers to the division of the cell's cytoplasm, including its components. So different organisms have different processes associated with cytokinesis. For example, in animal cells of the animal, cytokinesis begins with the formation of a cleavage furrow, so which initiates the division of the cytoplasm to produce two new cells. So plants, by contrast, will form a new cell wall that will eventually divide the two cells. So the cell wall material that is deposited during the cytokinesis in plants is known as your cell plate. So this is your cell plate. Once the cells have fully divided, the products are known as your daughter cells. Mitosis and meiosis have other important differences. One of these is the amount of genetic material present in the daughter cells. Mitosis produces cells with genetic material that is exactly similar in terms of amount and identity to that of the parent cell. So meiosis, on the other hand, produces daughter cells that have half the amount of your genetic material. So as what we have mentioned in our week 6 lesson, that the daughter cells or the chromosomes of the daughter cells in your meiosis is reduced. And this genetic material produced had some degree of variation compared to that of the parent cell. So this type of division allows the reduction of the chromosome number to form the sex cells. For example, if a diploid cell with 10 chromosomes undergoes meiosis, the resulting daughter cells will each consist of 5 chromosomes. Then the differences between the mitosis and meiosis will be discussed in the following slides. Okay, but then let us first remember that the processes or the process of mitosis and meiosis refers to the division of the nucleus and the genetic material, while cytokinesis, on the other hand, refers to the division of the cytoplasm. Now, cytokinesis can only occur once the genetic material is properly distributed to the daughter cells. We also have this what we call the cell cycle checkpoints. So if you look at all of the stages of the cell cycle, you will notice a general trend. So the cell needs to make sure that it's ready to divide, which it does so in the interphase. Some of the preparations, including duplicating the genetic material, acquiring nutrients, and doubling of the cell organelles. Once these preparations are done, the chromosomes are distributed to the daughter cell during your mitosis. Finally, the cytoplasm and other components are divided during cytokinesis. The cell cycle in general is an alternation between the duplication of your genetic material and the division of the cell. So imagine the times that you make a project for school. So what do you do when you make a mistake? Of course, you stop or you halt your progress in order to address the mistakes made. So if the mistakes is not too big, then you take your time to correct it. If the mistake becomes too big to fix, then you repeat your project all over again. So this bears some similarities to how the cell cycle proceeds. When the cell detects errors or improper conditions, then it halts or stops the progress of the cell cycle. It then tries to fix whatever errors are present. So if the errors are repaired, then the cell cycle continues. If these errors are too serious, then the cell may destroy itself. So this occurs through a process known as your apoptosis, or it also known as the programmed cell death. So the process of the self-destruction prevents errors from being passed on to the daughter cells. So there are multiple checkpoints present throughout the entire cell cycle. So these checkpoints are performed by specific protein molecules in the cell. So checkpoints include your G1 to S1 checkpoint or to S checkpoint. The next is to G2 to M checkpoint and the metaphase checkpoint. Both the G1 to S and G2 to M checkpoints occur during your interphase, while the metaphase checkpoints occurs during your mitosis and your meiosis. So by the end of the G1 phase, the cell cycle proceeds to the G1 to S checkpoint, or simply the G1 checkpoint. So this checkpoint makes sure that the cell is large enough to with all the necessary energy reserves and doubled organelles, and that there is no damage in the cell's DNA before the cell cycle continues. If the G1 to S checkpoint deems the cell to be unprepared, then the cell cannot proceed to the next phase. So if a cell is not committed to further prepare for division, it may enter a stage known as your G0 phase. So likewise, a cell in which the conditions of the G1 phase are not met will also enter the G0 or G0 stage. Thus, it is also considered an extended G1 phase. So the cell then may proceed with the cell cycle if the conditions become favorable for cell division to occur. So some cells, however, stay permanently at G0 phase, such as cardiac and your nerve cells. So the premise of the resting state for the G0 phase is that their activities are already outside the confines of normal cell cycle. So these G0 cells are not resting per se because they are still metabolically active. So the M phase, which involves either mitosis or your meiosis, will proceed after the G2 phase ends. However, the G2 to M checkpoint, or also known as your G2 checkpoint, makes sure that the cell is prepared before mitosis or meiosis. Some of the factors checked during this checkpoint includes the presence of your DNA damage, whether the chromosomes are properly replicated, or if environmental conditions are favorable for cell division to take place. If the DNA is properly replicated, no damage is present, and if environmental conditions are optimal, then the cell proceeds to either your mitosis or your meiosis. And the last stage is your metaphase or the metaphase checkpoint. It is found in metaphase during your mitosis. So it is in metaphase where the spindles are finally attached to the sister chromatids, which will then be separated during your anaphase. In order to ensure a proper and equal separation of these sister chromatids, the metaphase checkpoint will first check if the spindles are properly attached. The cell proceeds to anaphase if all spindles are properly attached. Remember, if the spindles in your metaphase are not properly attached to the sister chromatids, then eventually it will result to the cell cycle error, which includes your uh, meiotic non-disjunction. So otherwise, mitosis poses to remedy the error. Of course, we all have discussed on this cell cycle and the stages of your uh, cell cycle as well as the checkpoints. Now, let us proceed to the stages of your mitosis. So you have learned that the cell also undergoes its own life cycle. So the cell cycle describes the different processes that the cell undergoes throughout its existence. So animals and plants reproduce in order to propagate their species. So cells undergo a very similar process in order to produce more cells that are similar to them. This process is known as mitosis. So it is an essential part of the cell cycle. Without it, our bodies will not be able to grow and develop maturely or properly. So what is mitosis and why is it important? So what are the stages of mitosis and how do these stages contribute to the division of the cell? So have you ever wondered how humans or organisms grow and develop? And that is because of your mitosis. How about healed wounds? How does the wounded skin come back to normal? Or, yes, as we have mentioned in our basics lesson, there are scars in our body. So that is a result of this, what we call the mitosis. So the process of mitosis refers to how a cell, known as a parent cell, divides to produce new cells, known as your daughter cells. So recall that only somatic cells or the body cells undergo mitosis. Sex cells like the sperm and egg cells undergo a different process of division known as your meiosis. So take note that mitosis and meiosis both refer to the process of division that only individual cells undergo. So the role of mitosis in the cell cycle, it begins with interphase. The interface has multiple phases, which includes your G1 or your gap1 phase, your S phase, and your gap2 phase. The M phase follows your interface. So it is in the M phase wherein the cell undergoes mitosis if it is a somatic cell, or meiosis if it is a sex cell. Now, why is mitosis important for cells? So what are the different processes involved? So a cell will only begin the M phase if the conditions are correct and favorable for cell division. These conditions are largely affected by how the cell is prepared for mitosis by the different stages of interphase. So let us recall that the cell cycle checkpoints determine if the cell is prepared for mitosis or not. If the checkpoint deems the cell prepared, then the mitosis or the process of mitosis can continue. If not, then the cell will attempt to fix the errors, or it may undergo your programmed cell death, which we call the apoptosis. Now, the gap1 or gap1 and gap2 phases of your interface are highly similar in that they prepare the cell for division. So in multiple ways. So these phases involve the growth of the cell and the preparation of materials for cell division. So there are also cell cycle checkpoints that will check for proper growth after these phases. So proper growth of the cell before mitosis can occur. So this is because cell contents will be partitioned later to the daughter cells. So if the parent cells or cell does not grow properly or produce the necessary materials, then the division of cellular materials to be distributed to the daughter cells will also not occur. So the S phase preparation, or the S phase for the preparation of your mitosis, involves the duplication of the cell's DNA. So since mitosis aims to produce daughter cells that contain the proper amount of DNA, duplication of DNA is necessary beforehand. So more specifically, the parent cell aims to produce a similar amount of chromosomes for the daughter cells in mitosis. So suppose that the parent cell does not double the DNA that it has before mitosis. If this is the case, then the parent cell will only be able to give the daughter cells half of the DNA that is required. So doubling the DNA beforehand, however, will result in the proper amount being given by the parent cell to both the daughter cells. So this is the stage, or the stages of your the synthesis phase that involves the duplication of the cell's DNA. Recall that a cell's information is stored in the DNA, or the deoxyribonucleic acid. So DNA is in the form of chromatin in interphase, and it condenses into chromosomes during your mitosis. So as shown in the figure, those are the parts of your set chromosome. So each chromosome is made up of two sister chromatids. So the centromere connects the two hubs of the chromosome, and the centromeres have accompanying protein complexes known as your kinetochores. So the short arm and the long arm are the short and long ends of the chromosome respectively. So it is in these arms that the genes are found. So each end of the chromosome is known as the telomere. So these parts, along with the others, play significant roles in your mitosis. Since humans are diploid organisms, our cells have two pairs of each chromosome in our cells. So the human genome has 23 pairs of chromosomes, for a total of 46 chromosomes in each cell. 23 is the haploid number, whereas 46 is the diploid number. So haploid is often represented as N, or small letter n. A numerical coefficient is often attached to n to denote the number of copies of chromosomes that a cell or an organism has. For example, a diploid cells are represented as 2n since they have double the amount of chromosomes compared to haploid cells. So triploid cells are 3n, tetraploid cells are 4n, and so on and so forth. So therefore, if a 2n cell has 6 chromosomes, each haploid would have 3 chromosomes. So the number of sets of chromosomes that a cell or an organism has is known as your ploidy. So different organisms have different levels of ploidy. Humans, as was mentioned earlier, normally have diploid cells. So water bears are triploid. So salmons are tetraploid. So some species of frogs are dodecaploid, or with 12 sets of chromosomes. So each organism has a varying numbers of chromosomes. Now, after the cell has grown and manufactured the necessary components, mitosis can now begin. Mitosis aims to divide the parent cell into two identical daughter cells. So these daughter cells should have the exact same amount of chromosomes as the parent cell. So the daughter cells should also be able to receive the cytoplasm, the organelles, and other components from the parent cell. So in order for this to occur, the process of mitosis is made up of steps and sub-processes that allow the proper division of many components that the daughter cell needs. So much like interphase, the process of mitosis is also divided into multiple phases. So these phases are prophase, prometaphase, metaphase, anaphase, and telophase. So each phase performs a crucial role that contributes to this process of cell division. So the first stage is your prophase. In interphase, the genetic material is still in the form of chromatin. Once prophase begins, however, the chromatin will start to condense into compact structures known as the chromosomes. This process of condensation is important because DNA is normally a very long molecule. When it condenses, the resulting product is a small and compact chromosome that can easily be divided during your mitosis. So while the condensation of genetic material occurs, other events also take place in the cell during your prophase. One such event is the shrinkage, eventual disappearance of the cell's nucleolus. So another important occurrence is the formation of the mitotic spindle in the cytoplasm. The mitotic spindle is made up of proteins called your microtubules, and will be used for separating the chromatids later on in mitosis. So this process of spindle formation occurs in the area of the cell known as the centrosomes, of which the cell has two. So each centrosome further contains a pair of centrioles, and this helps with the formation of your mitotic spindle or your spindle fiber. Now, you also have this prometaphase, or before this cell proceeds to the next phase, which is your metaphase, a prometaphase will first occur. So it begins after the nuclear envelope or the membrane that surrounds the nucleus breaks down. So the two centrosomes, along with the accompanying centrioles and developing spindles, will move toward opposite sides of the cell, resulting in centrosome on each side. So each side is known as a pole. The spindles then start growing to the middle of the cell. It is also during this phase that the chromosomes, kinetochores, and centromeres become apparent. So this is important for the next phase of your mitosis. The next phase is your metaphase. So in your metaphase, the microtubules of the mitotic spindle attach the interact with the kinetochores of the chromosomes. So this causes the chromosomes to align at the center of the cell in an area known as your metaphase plate. So metaphase ends once the proper alignment of the chromosomes is achieved. So this alignment is when the sister chromatids of each chromosomes face the opposite poles of the cell. And the next phase is your anaphase. In your anaphase, the mitotic spindle pulls and separates the sister chromatids apart. So as shown here in the figure, the chromatids, now called daughter chromatids, are then pulled toward the opposite poles. So the movement will stop once the daughter chromatids completely reach the opposite poles. And the last phase is your telophase. In your telophase, or the final phase of your mitosis, after the daughter chromatids reach opposite poles at the end of anaphase, they will start to condense, deform, or they start to decondense. So the form that the genetic material will take returns to the one similar to anaphase. So the nucleus will also start to reappear. The nuclear envelopes will also reform. Once this is complete, there will be two entire nuclei with one found on each pole. So in animal cells, some of the events of the division of the cytoplasm will ensue even as telophase occurs. This process continues further in cytokinesis to become the cleavage furrow. In plants, on the other hand, the Golgi vesicles will start to accumulate to where the metaphase plate used to be. So this accumulation will allow the vesicles to fuse and form the cell plate, so which becomes your the cell plate. So both the cleavage furrow of animals and cell plate of your plants become the new boundaries of the two daughter cells. Now, when trying to differentiate between the appearance of cells in the different phases of mitosis, remember that the first letter of the phases shares the first letter of what the cell looks like in the phase. So let's have this one. In prophase, the cell's genetic material resembles polka dots or strings. In metaphase, the chromosomes migrate to the middle of the cell. And in anaphase, the chromosomes move away from each other. In your telophase, the parent cell starts to form two new cells. So always remember this one in your mitosis. And after your M phase or after your telophase, cytokinesis will divide the cell into two. Cytokinesis is not a phase of mitosis, but it is closely related to it. So recall from the what I have mentioned earlier that each of the two cells or two daughter cells will inherit a nucleus, similar genetic material, organelles, and more. So in humans, the number of chromosomes in somatic cells is 46. There are two sets of chromosomes, while sex cells have 23 or one set of chromosome only. So during your fertilization, it is the fusion of your egg and your sperm, each having 23 chromosomes, restore the number of an individual, which is 46. Therefore, if a cell before the S phase has 3 pairs of chromosomes for a total of 6, or there is a total of 6 chromatids, the 6 chromatids become 12 after DNA replication in the S phase. Do not be confused with chromatids and chromosomes. So here, let's have it this one. So to visualize the number of chromosomes and chromatids before the mitosis, after replication, and after mitotic division. So this is the reason why the daughter cells have the same number of chromosomes as the parent cell when it undergoes mitotic division. So we have learned about the stages of your mitosis and your mitosis. Now let us proceed to the stages of your meiosis. So sheep that belong to the genus Obis have 54 chromosomes. Just as previously discussed, this total number of chromosomes in a species represents two complete sets that are present in almost all body cells. So humans likewise have two complete sets with a total of 46 chromosomes. The primary mechanism of organisms to reproduce is through sexual means. And this process involves two parents. Now, have you ever wondered how the two parents involved in sexual reproduction are able to pass their chromosomes to their offspring while still maintaining the number of chromosomes for their species? Now, before each parent is able to contribute their genetic material to their offspring during sexual reproduction, some preparatory steps are also essential. In particular, the organisms need to produce the cells necessary for this type of reproduction to occur. Now, we will tackle how organisms use a process known as your meiosis for producing the cells that they need for reproduction. Now, like mitosis, meiosis also refers to how the cell divides to produce new cells. So sex cells or gametes are produced from the meiotic division. Meiosis is the M phase that leads to the production of either sperm cells or egg cells. This type of division is very closely tied with this process of sexual reproduction. Now, here's the question. How does the reduction of the chromosome number occur in meiosis, and why is this process important? Now, recall that the somatic cell undergo the process of your mitosis in order for them to divide. So a very similar process known as meiosis is the equivalent of mitosis to produce gametes or your sex cells. So in the process of sexual reproduction, a sex cell from a male, usually the sperm, and a sex cell from a female, usually the egg cell, will fuse together through a fertilization. Fertilization restores the normal chromosome number of the species. It will also lead to the formation of a single cell called a zygote, which will keep dividing through your mitosis and differentiating until it becomes an embryo. This embryo will further develop to become a mature organism. So if both sperm and egg have a full set of chromosomes, then the resulting zygote will have double the amount of chromosomes. This abnormal double amount of chromosomes will eventually be passed on once the zygote divides through mitosis and copied into the daughter cells. This can prove to be lethal or disastrous for the organisms. So to prevent this from happening, the sex cells or gametes of diploid organisms are usually haploid. So for example, a human normally have 46 chromosomes, but each of the sperm and egg cells only has 23. So the reduction of 46 chromosomes into 23 chromosomes in sex cells is possible through the process of your meiosis. So only sex cells undergo meiosis. As opposed to the process of your mitosis, the somatic cells go through. Many of the processes that occur in meiosis are very similar to the processes in the stages of your mitosis. So here is the stages of your meiosis. So your meiosis can be divided into two major phases, namely your meiosis 1 and your meiosis 2. So when interphase ends, the diploid precursor cell enters meiosis 1. So once the meiosis 1 is complete, the cell will then enter your meiosis 2. So recall that the phases of mitosis include your prophase, prometaphase, metaphase, anaphase, and telophase. So these phases are also present in each of the two rounds of your meiosis 2. So during the interphase, a cell duplicate, will duplicate its genetic material during the S phase. Once all the necessary checkpoints have been satisfied, then the cell that is destined to produce sex cells will enter meiosis 1. The cell that enters meiosis 1 will have the diploid chromosome number with 2 sister chromatids per chromosome. So this cell will eventually go through the different phases of your meiosis 1, namely your prophase 1, prometaphase 1, metaphase 1, anaphase 1, and your telophase 1. Now, here are the stages or the phases of your meiosis. In your prophase 1, similar to the prophase in your mitosis, the genetic material, which is equivalent to 2 chromosomes with 2 sister chromatids each, will start to condense. So there is a condensation of the chromosome. However, unlike mitosis, a process called synapsis also occurs in prophase 1. So synapsis is when each homologous chromosome pairs up and aligns from side to side. So each pair of the homologous chromosome is known as a tetrad. It refers to the 4 individual copies present in each complex. Once the chromosomes are fully paired with their homologs into the tetrads, the process of crossing over will then occur. So crossing over is when the homologous chromosomes exchange their segments with each other. And this happens in the chiasma. So during this event, the non-sister chromatids of homologous chromosomes exchange segments. Then after that, a recombination will take place. So recombination is the process wherein the DNA molecules are produced from two DNA molecules, or from recombination is the process wherein the new DNA molecules are produced from two DNA molecules or from different segments of the same parent. So DNA molecule. In the case of meiosis in sexually reproducing organisms, recombination is achieved through chromosomal crossing over. If the segments are exchanged, then the results will be chromosomes with new combinations of genes that contribute to the genetic diversity or the diversity of traits. Remember, in our discussion of your the application or the significance of your my cell division, we have discussed there that the crossing over of these chromosomes is very important. No, it can be seen in the traits of each of the siblings of your parents. The events of your prometaphase 1 are very similar to the ones in the prometaphase of mitosis. So the nuclear envelope will also disintegrate and the formation of the meiotic spindle will also occur. Recall that this process will help guide the chromosomes for migration later on. One major difference is that the meiotic spindle in meiosis 1 will guide entire tetrads and not just individual chromosomes. Now, the alignment of the tetrads in this middle of the cell will occur in metaphase 1. Again, this imaginary plane of the alignment in the middle of the cell, as shown in this picture, the aggregation site of the fluorescent chromosome is known as the metaphase plate. So once the tetrads have been properly aligned in the metaphase plate, the members of the homologous chromosomes will be separated from each other into the direction of the opposing spindle poles of the cell. So this process leads to the segregation of one homologous pair from its partner homologue in the tetrad. So the result of the migration is then a haploid set of chromosomes that migrate to each pole of the dividing cell. So the dividing cell will receive either the paternal or the maternal member of the homologous pairs, similar to this pair as shown in this picture or this figure. In telophase 1, the duplicated chromosomes finally fully reach the opposites or the opposite poles of the cell. So there are very few changes that occur in the chromosomes in this phase. However, the nuclear envelope may again form in some organisms, while it does not in others. So after telophase 1, cytokinesis occurs, which results in two daughter cells. Each daughter cell will have a set of haploid chromosomes. The cell then enters a stage called your interkinesis. So what will happen to the daughter cells if synapses and recombination do not occur? Okay, you try to answer this one on your own. And now, let us proceed to your meiosis 2. As what I have mentioned earlier, after the telophase 1, in your meiosis 1, the cell will enter into this what we call the interkinesis. So in your interkinesis, the meiotic spindles from meiosis 1 will disassemble and then reassemble in preparation for meiosis 2. So once this is done and other factors are fulfilled, then meiosis 2 can begin. So take note that both daughter cells from meiosis 1 can undergo meiosis 2. Meiosis 2 is more similar to mitosis than meiosis 1 is. So we have the first phase in your meiosis 2. In prophase 2, the genetic material will again start to condense and the meiotic spindle will again begin to form. The nuclear envelope will again break down in prometaphase 2. After your prophase 2, uh, we have your metaphase 2. The alignment of chromosomes in metaphase plate will occur in metaphase 2 with the help of the mitotic spindle. So take note that the tetrads align in metaphase 1. But since these tetrads have been segregated, only the duplicated chromosomes will align in metaphase 2, forming only one row similar to mitosis. The next phase is your anaphase 2. So, in anaphase 2, the meiotic spindle will start to separate the chromosomes in the metaphase plate into their sister chromatids. So these chromatids, which are now considered individual chromosomes, will migrate to the opposite poles of the cell. And the last phase is your telophase 2. In telophase 2, the nuclear envelope reforms the chromosomes decondensed, and the meiotic spindles disassemble into microtubules again. After cytokinesis, each cell that underwent meiosis 2 will have formed two new daughter cells. Thus, if there was one cell that underwent meiosis 1, this cell will have formed two daughter cells by the beginning of your meiosis 2. Each of these daughter cells would have undergone meiosis 2 to form two daughter cells each, for a total of 4 daughter haploid cells. So your mitosis and meiosis have key differences from each other. These differences lead to different effects and applications in the cells that undergo their respective phases. Now, do you still get the cells and cell division types mixed up? So to help you remember which is which, never forget the mnemonic device meso and mega. So meso means mitosis for somatic cells, and mega or meiosis for your gametes. Now, mitosis occurs in somatic cells. So these are the non-sex cells of the body. So the stages of mitosis are prophase, prometaphase, metaphase, anaphase, and your telophase. Meiosis is the type of division that produces sex cells or your gametes. It can be divided into two general stages, namely your meiosis 1 and your meiosis 2. So the stages of meiosis 1 includes, of course, your PMAT 1 or your prophase 1, prometaphase 1, metaphase 1, anaphase 1, and your telophase 1. Synapses and crossing over and the crossing over of the chromosomes only occur in your meiosis 1 and not in your mitosis nor in your meiosis 2. So meiosis 2, on the other hand, has different stages. So namely your prophase 2, prometaphase 2, metaphase 2, anaphase 2, and your telophase 2. So, mitosis results in two daughter cells that are genetically identical to the parent cell. So that's in a diploid organism will produce diploid daughter cells after your mitosis. So cell division only occurs once in mitosis. Meiosis results into two daughter cells after the first division of mitosis, or your meiosis, I should say. Each of these daughter cells will further divide after meiosis 2 for a total number of 4 daughter cells. So an example is given for the case of humans in this picture. Now, recombination occurs in meiosis, which results in each daughter cell having a different combination of genes compared to that of the parent cell. So in mitosis, it maintains an identical set of genetic material in the daughter cells, whereas meiosis reduces the amount of the genetic material and introduces new combinations of uh, genetic materials. So these have different consequences and application as a result. So some of the differences between the meiosis 1 and your meiosis 2 are the following. So synapses or the organization of the chromosomes into complexes of tetrads occurs only in your meiosis 1. So crossing over between homologous pairs occurs only in your meiosis 1 also. Tetrads line up and separate into individual homologs or homologous chromosomes in your meiosis 1. So the duplicated chromosomes or the sister chromatids line up and separate into individual chromosomes in meiosis 2. Now, crossing over, as was explained earlier, occurs in homologous chromosomes during your prophase 1. So this process involves the exchange of segments between the two homologous chromosomes. This exchange of genetic material between the two chromosomes will result in the formation of new gene combinations that are different from both of the parents. So this is especially important since it creates genetic variation in that the resulting daughter cells will have different genetic content from the parent cell. So the use of these daughter cells later on in the fertilization will further increase diversity. So that is why you look different from your siblings. Even if you are twins, there are still differences because what we have mentioned, this is because of the crossing over of the chromosomes during your meiosis. In meiosis 1, meiosis 1 begins with each chromosomes being found in pairs. So each having two sister chromatids. Note that at the start of your meiosis, there are 6 duplicated chromosomes or chromosomes with sister chromatids. The diploid number of which is given by 2n equals 6. This is the chromosome number that will be reduced into haploid as meiosis progresses. So meiosis 1 entails the separation of each of the members of the homologous pairs of chromosomes after they have paired and crossed over. So that after meiosis 1, the starting diploid chromosome number of 2n equals 6 is reduced into n equals 3. So the chromosome number is already reduced into haploid. However, it is still not normal for cells to have duplicated chromosomes after division. And in this case, the products of your meiosis 1 are 2 haploid cells with 3 duplicated chromosomes. The primary goal of your meiosis II is to separate these sister chromatids of the duplicated chromosome. Both of the resulting cells from your meiosis I with n equals 3. It enters meiosis II. Note that in the second meiotic division, there is no further reduction in chromosome number. Its only purpose is to separate the sister chromatids, similar to the mechanism of mitotic division. By the off meiosis II, every cell gives rise to two more cells with haploid chromosome numbers, each consisting of three single chromosomes. Note that the apparent difference between meiosis I and meiosis II in terms of the change in chromosome number. Meiosis I is a reductional division because you start with 2n equals 6 and end up with n equals 3 cell. By contrast, meiosis II is an equational division because you start with n equals 3 and end with the same chromosome number. Now, let's sum it up. Or let's have a review, let's have a recap. So the life cycle of a cell is known as the cell cycle, and it describes how cell grows, develop, and reproduce. With a very few exceptions, all of the cells in your body contain genetic material in the form of DNA or your deoxyribonucleic acid, which is usually found in the nucleus of your cell. So there are three main stages in the cell cycle, namely your interphase, your M phase, and your cytokinesis. Your interphase is the stage when the cell prepares itself for eventual cell division, which can either be mitosis or your meiosis. It is also the stage when the cell is metabolically active and performs its specialized function. Cytokinesis refers to the division of the cell's cytoplasm and other components. So this process significantly differs between animal cell and plant cells, primarily because of the presence of the cell walls in the latter. So here are the stages of your cell cycle. It includes your interphase, and in your interphase you have your G1 phase, you have your S phase, and you have your G2 phase. After the interphase, you have your M phase, and after your M phase, lastly, you have your cytokinesis. You also have these stages of your mitosis or your M phase. Interphase first, of course, then mitosis. Under your mitosis or your M phase, you have your prophase, prometaphase, metaphase, anaphase, and your telophase. After your telophase, you will have your cytokinesis. The process of sexual reproduction requires a reduction in the chromosome number. This is achieved through meiosis, the type of cell division that produces gametes or sex cells. Unlike mitosis, meiosis involves two rounds of division, namely meiosis and meiosis II. Meiosis II involves or immediately takes place after meiosis I, so only the latter is preceded by the duplication of your genetic material. Aside from the number of daughter cells produced, meiosis is also different from mitosis in terms of its unique events. One of which is your synapsis, which involves the pairing of the members of homologous chromosomes so as to allow them to undergo crossing over and to segregate them. Aside from the number of daughter cells produced, meiosis is also different from mitosis in terms of its unique events. The crossing over refers to the exchange of segments between non-sister chromatids of homologous chromosomes. Ultimately, this enhances the genetic diversity of species. Meiosis I is considered the reductional division in meiosis because it reduces the number of sets of chromosomes from 2 to 1. Meiosis II, by which contrast, is considered the equational division in meiosis because there is no further reduction of the chromosome number in your daughter cells. In meiosis I and meiosis II, they are also different in terms of the segregating elements. Meiosis I entails the separation of the members of the homologous chromosomes, whereas meiosis II involves the separation of sister chromatids. Meiotic division can be described according to the number of divisions, number of daughter cells, and the unique events that take place. DNA is the form of chromatin in interphase, and it condenses into chromosomes during mitosis. So these phases are prophase, prometaphase, metaphase, anaphase, and telophase. Each phase performs a crucial role that contributes to the process of your cell division. Chromosome condensation happens in prophase. Spindle formation also begins. In your prometaphase, begins after the completion of the breakdown of the nuclear envelope. Metaphase involves the alignment of the chromosomes in the metaphase plate. The sister chromatids move to the opposite poles of the cell in anaphase. Telophase ends when the two nuclei are formed in the cell. So that is all about your cell cycle, the cell division, mitosis, and your meiosis. Okay, so before we wrap up, let us answer this following question. I want you to answer it on your own.